Hi, my name is Matty Hans, and this summer I worked here at Riverdale at the Summer Science Research Program, focusing on environmental toxicology of the Bronx River. Specifically, we focused on Spartina alterniflora, which is a native organism to the river. It's a smooth cordgrass, picture of it here, that grows in estuaries. And so the purpose of our experiment was to monitor the environmental impact of the Bronx River compared to our relatively clean site in Greenwich Cove, Connecticut. And from previous summers, we've made some observations, one of which is that the Bronx River had comparatively low dissolved oxygen levels, which negatively impact the organisms living in the river. We also chose to add Spartina alterniflora to our indicator species to broaden our spec of indicator species, along with geomissile mussels growing in both sites. A question we posed was that if Spartina serves as a filtration system for the mussels living on the grass, if that increases the stress levels. And our approach was to monitor the expression of stress-induced biomarkers, specifically shock protein 70 here in the marsh grass. Here's some pictures of the Bronx River, and as you can see, a lot of metal pollutants and contaminants in the river. This is the Spartina living in its environment. So heat shock protein 70, it's found in all stages of life from bacteria to humans, so we knew that we could find it in the grass. It's present under all conditions, but then when it's induced, it's expressed more because of organisms exposed to higher stress. The main function we focused on was that it acts as a chaperone protein for protein denaturation in non-stress cells. Here's HSP70 working with a protein that has been unfolded, so it's pretty much denatured. And it works with ATP chemical energy produced in the cell, and it uses ATP to correctly fold that protein and make it so it works properly. Our specific experiment was that we took control samples of grafts from both sites, and we incubated them at room temperature, 26 and about 27 degrees Celsius, and then we took separate samples and we heat shocked them at significantly higher temperatures, and this was to max the stress levels of HSP70, and then we extracted protein and then determined the concentration of the proteins, and then determined the levels of HSP70 through Western blot analysis. Western blot analysis was done through electrophoresis, which takes a specific molecular weight, specifically 70 kilodaltons, which stands for the 70 in heat shock protein 70, and you you transfer the gel to a membrane so that you can take a photographic copy of that sheet, so that you can quantify the intensity of the pixels, and that's the Bronx heat shock, Bronx control, Greenwich Cove heat shock, Greenwich Cove control. And what you can do is, you can take the intensity of those pixels and graph them so you can visualize them. On the y-axis, we have the intensity of those pixels. On the x-axis, we have the different types of Spartina analytes. The green is the control, and the red is the heat shock. And we found three findings that suggest from this. One was that the controls were a lot less stressed than their heat shock. Also that the Bronx organisms were under a lot more stress than those of Greenwich Cove, and also that the roots were under more stress than those of the leaves, which we found suggests that because of where the roots are in their environment, that the roots are always underneath the soil compared to the leaves, which would only be stressed at high tide. And so I'd like to thank Dr. Cox, who mentored me, and then people who funded our research. Thanks.